one of the folks at UCSF who's running the EQUIAX study. We're currently enrolling at San Francisco General, our county hospital. And we are hoping to open some new sites, but currently are facing some funding radio silence from FDA. So we've got folks on this call from the team that's running things at San Francisco General, but also people who are here who we're really hoping will be launching new sites for EQUIAX. And then I see a lot of familiar faces for people who I know have either been enrolling folks or about to start enrollment. So Mike and I were thinking maybe we could just open the meeting by going around and giving some updates about anything that's going on at your site in terms of enrollment, if it's happening, if it's about to happen, and if there are any barriers that we can work on as a group to help troubleshoot. So I think maybe we can just sort of, I know all of our Zoom projections are slightly different, but maybe Brady, do you want to give an update from Rush, and then we can kind of hand the mic around to the group?
Sure. For those that may not know me, my name is Brady Scott. I'm a respiratory therapist and associate professor at Rush University in Chicago. We currently have an IRB approved study that actually has sent to Carolyn and Mike and the group to take a look at our protocol to see if we were in general alignment with everybody. Carolyn made some excellent edits that we have made, and I have since then even met with our statistician to rethink our enrollment numbers. We took a shot at it once, but then I wanted to get an actual statistician on board to make sure that we were close. And it turns out we were actually pretty close once we had several discussions, but I think I have a better number. Looks like we're going to try to target about 140 individuals for our study based on their recommendations. Our current study was just looking at using the Monk scale. However, I'm happy to report that as of today, as a matter of fact, ironically today, I have full approval to spend all kinds of money on buying the Konica Minolta device. And I'm just working with my finance guy to take it out of my budget and a little bit of another budget and put them together and to be able to make that purchase. And actually got on the phone with my IRB representative today just to discuss putting that through, what that needs to look like, and to make sure that we're still under the same categories. And so everything looks good in terms of getting that ordered. I've got a new quote for those of you that are looking to buy the device. My initial quote for the device came in January, and I got a new quote and went up by about, I don't know, somewhere between 5,000 or 500 and maybe $1,000. It went up a little bit from the initial quote, even with the discount. So the cost went up a little bit, but hopefully we'll have that device in hand. So it looks like our study will include both the Monk scale and the Konica Minolta device for those measures. And then once we get that, we'll be off to the races.
Congrats. That's really a great update.
And thank you for your help along the way. I know I've sent you 1,000 questions and things. I'm just making sure that we're all in alignment, that we have useful data at the end of the day when we need it.
Yeah, no, thanks for letting us be involved in your study development. And that's, ultimately what we're interested in too. And one of the things that Mike's going to talk about later in the meeting is just to make sure that we have a general agreement about data sharing and an idea of how to make the biggest impact with the data where each group is collecting. And of course, everybody's going to have, their particular area of interest, but there are things that we're all working on together. So thanks for allowing us to work with you. Mike's got his hand up.
I was just going to comment really quickly, since the Konica came up, I'm sure this will come up with other folks too. We continue in our study to try and figure out the best way to use that data, but also to collect it. There have been issues ranging from like the actual application of the device on different body parts to just some like nuances to using the software with like issuing some experiencing some like data capture problems that we're noticing while we're doing our prelim analyses. I hope to be able to report back more specifically on some of those things, but maybe we can talk about it later if there's time. But Brady and others who are using the Konica might be good to loop you in on some of our discussions that are much more focused on the Konica. Today, I'm going to be in the lab. We're going to be troubleshooting a few things that we're having, and then we're going to do another prelim analysis very shortly to try and work out some of these kinks, but hopefully save you some time. Also would love input from the experience that others are having with the device once you're using it.
Great. So I
Don't
know
worry, it's worth buying. It's working. We're just, it's like very small things.
It's an important follow-up. I see there are multiple people from Rush on the call. Is there anyone else who wants to add anything to what Brady's shared? Okay. All right, maybe Lale Jalilian from UCLA, who's already been enrolling patients, could give us an update on how things are going at your site.
My name is Dr. Lale Jalili, and I'm one of the anesthesia intensivists here at UCLA. I practice primarily in our cardiothoracic ICU. I have been collaborating. I actually got into this project through an engineering lab at UCLA that primarily approached me because they were interested in a technological innovation and redesigning the pulse oximeter. So we approached it not to really from an engineering angle. I met with your study coordinator, I think a month ago, and apart from the Konica Minolta, it sounds like he and I have been experiencing the same, challenges in terms of getting the data. But I am continuing to enroll in both our cardiac cath lab, which is a very controlled environment with patients who have known hypoxemia just because of congenital cardiac disease. And I also have been collecting data across all 8 of our ICUs here at UCLA with patients as young as our neonatal population in the neonatal ICU to pediatric. I'm approved for like all age ranges. I think the primary challenge that I've been having over the past few weeks is, at least from an engineering standpoint, as a team, we're not sure that the initial innovation that we thought might work will work. And so because of that, I've actually paused on data collection for now as we kind of assess as we assess more the technology that we've been developing and seeing if it's actually feasible or not. I think the data that I do have though is like almost identical to the way that UCSF is collecting it, so it would be very easy for me to share. We've also been capturing raw waveform data, not just like the actual number, but we have raw waveforms that would be interesting to look at. I think like more broadly, I had been hoping to use this preliminary data to get a grant, at least from an engineering standpoint, like proposing an engineering innovation. I need to think about whether that's going to actually work and whether like the premise of our proposal is going to work. So I think that's where I am right now.
Lale, quick question. Hey, Mike from anesthesia. I know we've been on emails before. Great to see you. I had a quick question. In terms of your raw waveform, are you capturing like processed raw waveform, like the visual waveform, or are you capturing unprocessed wavelength specific AC/DC current? Like, how are you capturing it?
I think it's the latter. Sorry, I need to ask the grad students who there's like a team of grad students who came up with this setup and they just gave it to me. So we have a novel pulse oximeter, like our own custom designed pulse oximeter, and that's capturing its own waveform. The hardware is exactly identical to like a standard pulse oximeter, but these students have made some modifications and we're like analyzing that data right now. But we've also been getting raw waveform data from the not the Masimo, not the Nonin. Sorry, I'm blanking on the third commercial brand. Which one was it?
Nellcor?
Sorry, which one?
The Nellcor?
Yeah, I think we're getting raw waveform data from there. The students couldn't break into the Masimo. They were close. Yeah. We just bought a Masimo pulse oximeter. They just like opened it up and like go in there. So, but they weren't able to figure it out for the Massimo. But I think for the NEL core, we have raw waveform data, and then from our own custom pulse oximeter, we do.
Just to flag, and we can try and link you in with this, we have like a data team that's just doing raw waveform analysis and device creation. We're happy to invite your engineers to join as well. Some of the members of that team are former engineers for the major companies.
Oh, really?
You know, obviously, yeah. So I'll leave it at that. But yeah, we're happy to include you. We also have a device that we have made. We're about to build version 2 of it that captures raw signal and has multiple wavelengths and things like that. And we're hopefully in the next probably like 2 to 3 month timeline, we'll be pushing all of our scripts for data analysis and like waveform analysis into a new Open Oximetry GitHub account. So if you have, again, like your engineers and stuff, we'll be able to kind of hopefully get people to start sharing what they're doing with the waveform analyses. But yeah, let me know, maybe send me an email separately and link it with your team, and we can try and loop you guys in if interested. Okay, the data repository that I'll talk about later will have the ability to capture raw waveform data. And so we're speccing out like what the characteristics of that would need to be to contribute to that database. So it'd be great to kind of learn what you guys are up to and compare notes.
Yeah, we have a lot of raw waveform data. I've been playing around with it too. But yeah, that's where we are right now down here.
And Lale, I know you've been sort of leading the charge and doing a lot of work hands-on yourself in the OR. And I'm wondering if you could give us an update on your enrollment numbers to date. I know your population spans the breadth of ages, but how many folks have you been able to enroll for your preliminary efforts?
Yeah, since we last talked, I actually stopped. I'm only up to 33. The primary reason is because it's very time consuming to get this data. And unlike the rest of the people on this call, I don't have a grant. So I actually am a little bit concerned, like if we're not going to have like some innovation or some technological innovation that we want to pursue, like as a full-time clinician, it's really hard for me to get this data. Like even with the support of my chair and like even going to our OR schedulers, just to like get 30 minutes, like get out of the OR to go get this data. I mean, I don't have a study coordinator. So it's really hard without someone who's actually full-time dedicated to do this. And, you know, as a group, we're approaching it more from an engineering, standpoint. So I kind of paused collecting data just to kind of step back a little bit and see like what is the direction that we as a group want to go. That's feasible for me also.
I'm impressed you've done as much as you have without CRC. It's unbelievable.
Yeah, agree. That's a big lift. Okay, well, thanks for that update, and let's see if maybe there's a connection with the engineering students that might be helpful to keep your project afloat.
I put something in the chat, but I just wanted to point out to the group that there is an NSF program that I think would be appropriate for this work from the engineering perspective, as you said, Leila, that you would need to have some technological innovation, device kind of development, but also with the data science connection. If you're not aware about it, I put a link there, and I'm simultaneously working on some preliminary work to send in a call in November if anyone's interested in talking more.
Yeah, thank you so much. I'm going to take a look. I'm basically looking at any like grant funding I can find at the moment. But also, these projects from the very beginning, they need to make sense, I think. So yeah, thank you so much for this link.
Yeah, thank you, Neil. Is there anything while we are chatting, is there anything that you would like to give an update on from St. Louis?
Yeah, I don't have a study that's going on, but we are still looking at data sources and doing simulation essentially to try to validate our hypothesis, which is that this method that we use to take the raw data, the algorithm that takes this ratio of these different components in order to estimate the SpO2 value, that algorithm introduces some of the bias into the measurements that are made. So we've been running simulations to show that this turns out the way that that we expect from the statistical side, and I don't have anything in particular to report because I'm hoping for data from studies that would allow us to validate this with patient data versus the simulations we've been doing.
Got it. Thank you. And thanks for putting the link in the chat as well. Appreciate that. Sure. Okay, maybe Eric McCollum calling in from, are you in South Africa right now?
Yes, hi everyone.
Hi.
So for those of you I haven't met, my name is Eric McCollum. I'm a pediatric pulmonologist at Johns Hopkins, but based in South Africa. We have a project called Phefumla that where we're developing and validating a mobile phone reflectance pulse oximeter. And so we're nearing hopefully the testing phase with this device. And so there's multiple components of this project. Probably the most relevant aspects to mention here is that we have a pediatric pulse oximeter clinical accuracy platform that we're setting up at Tygerberg Hospital here in South Africa. So we're pretty close to getting that IRB approved, and that'll kind of serve as a platform for us to do clinical accuracy testing. So we would be pairing oximeter measurements of various oximeters with arterial blood gas measurements that are obtained in the pediatric intensive care unit. What's maybe a little bit different from some of the challenges I think many of you have been facing is that they do arterial blood gas measurements routinely in the PICU here. And so, we haven't yet demonstrated this, but we feel relatively confident that we'll be able to pair the measurements fairly well. I'm sure there'll be challenges, but that's the notion at the moment. And then we have a clinical trial that will be beginning early next year. We're in the final phases of getting IRB approval for that. That's a 3-arm randomized trial that is essentially implementing this particular reflectance oximeter, plus there's a device called the ACARE device, which Lifebox distributes. We developed a universal probe that is used on children, infants and children, and we've published on this, but it hasn't been implemented, and so that's something that that's going to be one of the arms. It's a clustered trial, and then the comparison arm will be the current pulse oximeters that are available. We're going to be doing it at 18 primary health clinics in the Western Cape of South Africa. So that'll be starting kind of early next year. So that's my update.
You have a lot of projects ready to launch. That's really exciting.
Can I ask a quick question, Eric? Do you know what blood gas analyzer are you guys using in your study sites?
Putting me on the spot. So this is, I'll have to double check on that. It's a brand that I'm not sure is used in the US. So I'll circle back to you on that.
Okay, thanks. That would be great. Part of the reason I asked, we've been exploring some data collection in a couple other countries in Africa, and some of the oximeters will do like a calculated, not measured functional saturation. but it's not so clear that that's what they're doing, and that data aren't as useful or just aren't useful. So just flagging that.
All right, in the pediatric research group, I don't see anyone from Stanford. And I saw April Edwell had joined momentarily, but I think had to leave. Am I incorrect? Is there anyone else representing the pediatric folks on this call? Okay, well, maybe we can go to another Johns Hopkins researcher. And Ash, you want to give us an update if you've heard anything about what might be happening at Johns Hopkins in Baltimore?
Sure. So, hi, I'm Ash Fawzi. I'm an adult pulmonary and critical care at Johns Hopkins. So, I had previously conducted a short study in adult NICU patients using the Konica Minolta. We recruited 12 patients, but it's been stopped for a number of months now, and looking for funding to restart basically, and hopefully be another site for the UCSF study. But no, I haven't heard anything back yet. So we're still waiting for word on whether our site is going to be funded or not. But without that, I mean, as Lale said, it's very difficult to conduct this study in any way without, dedicated funding. So at this point, we're still waiting.
Yes, and we have more colleagues who are also waiting. I see Farzad and Lance have joined from Highland Hospital, which is a county hospital in Oakland, just across the bay from us in San Francisco. And we are hoping to expand our enrollment to their site. I don't know if you guys want to give any updates. I'm up to speed and can do it if you would like to defer, but we're happy to have you introduce yourselves and say hello to the group.
I'm Farzad. I'm one of the ICU docs over at Highland, moved to the sunnier side of the bay. So yeah, I mean, we're still very interested in working on getting the IRB, everything set on that front. And I think we've sort of done the population screen already, and the numbers would look good from that standpoint to be helpful. I don't know if Lance has anything to add.
Hey everyone, my name's Lance Smith. I'm the clinical research associate at Highland Hospital. I've been asked to help get this up and running, and we're really just in the startup phase. We're waiting for our IRB to kind of approve our consent language and kind of sign a draft alliance agreement and get that ball rolling, and just getting people in the ICU aware of what we want to do, and kind of open up the possibility of having outside coordinators come and work with us. So it's a lot, but we've got all of the plates spinning right now. It's just going to take a little bit.
Yeah, and we're waiting on the FDA's funding decision as well to make sure that we can expand our CRC workforce, because as you've heard numerous times, this is just sort of not impossible for Lale, but impossible for most people to run without a CRC.
Can I add one quick thing on that, just for context? Yeah. If people are wondering, so we are recognizing, as everybody kind of knows on this call, answers to this question around skin color, but also likely there are other factors that are impacting pulse oximeter performance that we're going to want to dig into. It's going to take big data sets, and it's just not possible for one site to do it. And for all the reasons you're well aware of, it doesn't really make sense to collect data only in one site anyways. But the reason that we're specifically reaching out to Hopkins on this FDA to try and partner and expand with Ash and the team there is that Hopkins and UCSF are both CIRSE Centers of Excellence in Regulatory Science and Innovation, as part of an FDA network. There's like 6 of these in the country: Yale, Mayo, Stanford, Hopkins. And so that's why we are working most closely with Stanford and trying to expand and partner with Hopkins. So for those of you who are wondering, there's a very specific reason why we're talking about that, but just wanted to flag that. The CIRSE is a mechanism for funding. We weren't really as familiar with it before this, to be honest. But for those of you who are or have collaborators at those institutions, something to think about down the road. So just wanted to give that context.
Thanks, Mike, for connecting the dots. Okay, we have a few more sites represented. Mike Ruffo from PATH is another international investigator. Maybe you could give us an update on what's going on. I think you have some randomized control trials that you're working on.
Yes, hi. Yeah, we have two studies going through this project. One is a rather large study that's happening that is not necessarily looking at skin color, but has a large data set that could contribute and is looking at the impact and feasibility of implementing pulse oximeters in low and middle income countries, specifically on children under 5. And so that has two components. One's a randomized control trial that's being run in India and Tanzania. And then another component that is operational research and a pre-post study that's being conducted in Kenya and Senegal. And we've stopped enrollment, so we've finished enrollment. And the last follow-up happened in April, which is great. And those studies have some fairly large sample sizes or enrollment. So the Tanzania study has 108,000 kids, and India has 48,000 enrolled. That's for the RCT. And then the operational or pre-post study that's happening in Kenya had 31,000 and Senegal was 20,000. So some big data sets there. And really, I think the reason for that was because hypoxemia is not a incredibly common occurrence. And so we needed large data sets to understand. But with that comes a lot of data around hypoxemia prevalence and a lot of that. So those databases have been locked, and our partner Swiss Tropical and Public Health Institute is leading that work with research institutes in each of the project countries. So PATH isn't conducting the research, but we're sort of leading the whole project. The other piece, and so we're right in the middle of the analysis right now, and we're really excited to hopefully have some results coming up later this year. The other piece that is a little more related to the work being done in this group is a diagnostic accuracy study that's being done on a number of different devices that measure health indicators using the plethysmograph. So some what we refer to as multimodal devices. So looking at those and how they contribute or how they compare to a gold standard in just 2 facilities in each of the 4 project countries, and much smaller sample sizes there in the hundreds as opposed to the thousands. And so that is underway. And the other component of that will be an implementation study. So looking at how those devices function in the hands, or one of the devices at least actually is used, and some of how it affects the clinical processes and the health pathways for the children that are being diagnosed. So more of an implementation study piece. But first, the diagnostic accuracy study, which is underway. So I'll stop there.
And Mike, just to add on, we're excited to partner with PATH as part of the Open Oximetry project. So we're partnering with PATH on some of the lab-based work that we're doing here at UCSF, and some of the advocacy work that we hope will come from this, and then also some of the raw PPG data device capture stuff. So we're just sort of getting started with some of that work. So excited to be working with you guys.
Yeah, same. And thanks for providing the PPG waveform devices. And looking forward to really getting that underway. So thanks.
Sounds
Congrats. Good. Congrats on completing enrollment. That's really exciting, and we look forward to hearing about the data analyses. There's a few people who are writing in the chat that they need to hop off, but so I think we can kind of move through. We have 2 more sites. I think Mount Sinai and Vanderbilt are the other sites here. And then Mike wanted to share a data sharing agreement document for feedback and discussion. So I want to make sure we get to that. But anything new happening at Mount Sinai?
So last time we had a meeting, I mentioned that I was applying for an Anesthesia Patient Safety Foundation grant. So I had been invited to provide a full grant proposal, and I submitted that on Friday. So that's in. I should hear back by October, and it would be starting January 2024. So hopefully the Konica device doesn't go up too much between now and then. If I get it, we'll have to take a look and see. But that would be a prospective study in a pediatric cardiac anesthesia type study.
Congrats, Garrett. That's really exciting. And when do you hear back about funding?
In October.
October. Okay, great.
Yeah.
All right. And then Joey Starnes from Vanderbilt is also here. Any updates from you?
No, just thanks for letting me join the meeting today. I'm Joey. I'm a second-year pediatric cardiology fellow at Vanderbilt. And I recently had a T32 funded around this type of work, partially to look at the policy effect of critical congenital heart disease screening at the state level. So not really prospective data collection, but a second aim around prospective calorimetry and pulse oximetry data collection in our pediatric cardiac cath lab. And so still working on the protocol development for that, but have some funding from the T32, and the division is super supportive of equipment costs and stuff like that if needed. And so still working through some of that, but happy to be here.
All right, thank you for the update. And did I miss anybody? I think I've included all institutions, and there's a lot of people from UCSF here. All right, Mike, do you want to screen share the document, or lead us through the, oh, there you go. There's a Google Docs link.
Sounds good. I just put a link in the chat to this document. I'll give you some of the background, and then would love to invite people to add comments to the document. The objective of what we're trying to do here is, again, try and create a mechanism where we can pool data so that we can ask bigger questions, especially for groups that are collecting raw waveform analysis, a lot of physiologic data. And that isn't necessarily specific to just pulse oximetry. We anticipate a lot of people in the same studies will be collecting data on respiratory rate monitoring, non-invasive hemoglobin, things that are evolving, potentially impacted by some of the same issues that pulse ox are, and also highly relevant to resource denied and resource variable settings. And so that's kind of what our aim for this data repository is create a place where people can put their data. We're also doing this sort of in the spirit of the new NIH policies around scientific data sharing that are basically requiring any NIH-funded researchers to do this, to share their de-identified data sets in the public domain so people can use them for other purposes. And we think this is a great project, a great example of how we could do that across different sites. But instead of everybody creating all these different public data sets, if we can figure out a way to structure the data and merge it, that it would be a lot more powerful. And we have a little bit of funding to try and create that data set and clean and maintain it for the next 12 months. And so we're hoping that we can do like a pilot basically with any groups that are interested in doing this. The rough idea is you collect data, you publish it, you do whatever you would normally do with it. But then when you're ready, you would also put it into this data repository. We would provide a data dictionary and instructions on how to format your data and potentially even help you with some of that, depending on what you need. Then the data would go into this repository. The repository would be open access, but there would be request step. So people would have to demonstrate that they have done, for example, like city training and sign an agreement that says, we're not going to sell the data. There's precedent for this with the PhysioNet at MIT and a few other open data repositories. So we're kind of following that model. We may end up hosting it directly on PhysioNet or basically our own version of that. The decision tree there mostly has to do with whether or not PhysioNet would stay free. We want to make sure this stays free. And so we're just trying to navigate that. But if you're curious, PhysioNet's a great example of what this will look like. The document I shared is again, modeled off of the PhysioNet data sharing agreement and some of the NIH data sharing policies, but this is sort of what we're aiming for. We would love feedback on, whether or not you guys have been involved in any projects that have done this already that we can look at to try and take best practices from. Also, if you've done this in any of your other projects, IRB wording, how your institutions, what they require. Everybody's a little bit different, especially when there's international data. So we would love input on how to make sure we're not walking into any traps with this and get out ahead of that to make it easier. And then, for IRBs here at UCSF, we basically put in a line that's at the bottom of this document that basically just says, de-identified data from this project may be used for other analyses in the future and will be maybe shared in an open data repository in compliance with NIH data policies. And that has sufficed to cover us to be able to do this. But again, every institution might be a little different. This is sort of, I think, new for many IRBs and evolving. So we would love input. So with that, maybe I'll pause and see if anybody has questions, and the feedback, certainly welcome any at this point, but definitely can happen via comments in the Google Doc or via email. And then also, if nobody has comments on this specifically, would love to just know, I think some of you have reached out and said you're generally interested, but just kind of, if you could chime in on here, if you're interested, and then also like any immediate perceived barriers that you think we should start trying to address.
I'm sorry, Mike. I had somebody come in my office during, I think probably the biggest thing I wanted to hear from you was, is this data sharing?
Yeah, data sharing. I was just saying, I put a Google Doc in the chat for comment, but then we just want feedback on, is your team interested in this? What perceived barriers do you envision so we can start trying to address those?
And yeah, maybe I can, plant a seed. Sorry, Brady. Maybe I could plant a seed also for this discussion. If you are making a biorepository, so if you're keeping any of the blood samples that could have DNA analysis done on them, that's like a whole different ballgame with the IRB. So this language suffices for our IRB because we don't keep any of the blood, we just keep the data from the ABG results. But if you are keeping any tissue and thinking about looking at not RNA, but DNA, we'd have to talk about some other wrinkles there.
Yeah, I'll start, Mike. I think you already know we're interested, and I think this diagnostic accuracy study that I described, where we're using the waveform devices that you all provided, definitely want to contribute to the broader data set with that. Yeah, I think you had mentioned it briefly. Just one of the challenges we see is the different rules and laws that come into play when you're working in different countries. And so that's something we're trying to navigate. I think India has probably been the most challenging for us. So I think we'll continue to work through that, but that's the first thing that comes to mind when you think of challenges. I'll stop there. It's not going to stop us.
Appreciate that, Mike.
Yeah.
Ash, do you know of anybody at Hopkins who has an open data repository, like NIH funded folks that are complying with that new requirement?
Not specifically off the top of my head. I mean, we do have our repository here where you could put data that Hopkins approves of and the IRB approves of, obviously. But I think in terms of we're part of some multi-center studies separate from this where the data is shared on like BioLink and things like that through the NIH, and that's not been a problem either.
Okay. Any other comments from folks? Eric, is your IRB going through Hopkins or a local institution in South Africa, or both, or many?
It'll be both. It'll be both. Yeah. I mean, I'm reflecting on what Mike said, and I think I need to kind of look a little bit further into the data sharing aspects here in South Africa, but we will overcome them. we're interested, of course, in contributing.
Sounds good. And, one of the things that has come up is that, every group's likely to have their own database. It may be the case that, if each group creates their own de-identified data set and disseminates that in a way that is allowed by their local IRB and basically familiar to their local IRB, whether that's an open access version of the local institution's REDCap that is de-identified and blah, 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 or, through an existing mechanism that we take that route, everybody puts their data into whatever is allowed by their institution, and we will go and figure out how to bring that together into a unified source. That may be easier than saying we're going to put it in this new data repository and all the IRBs are going to say, what the hell is that? We've never heard of that. And so that is a possibility as well. We're totally open to it and have been talking about strategies where we could try and help automate that process a little bit. But that's, just flagging that. Brady, were you going to say something, sir?
No, I've just been interrupted almost the entire time I've been in this meeting, so I apologize.
It's all good.
I got a lot going on in the background here, so I'm just phones ringing, all these things, so I apologize.
We know how it goes. Well, since you guys are getting ready to start your data collection, please let us know, how we can be helpful. And also, as this data sharing piece comes up, we would like to, we're probably still a couple months before we really start dumping data into it. And we'll have another meeting of this group before that happens, but would love to launch it, ideally with partners and collaborators and not have this, our intention, we're not going to be branding it as UCSF. We would brand it as the Open Oximetry Project, and would love to do that with, in collaboration with groups that are contributing data and all that. So, as you're thinking also about applying for other funding, hopefully that can be leveraged to improve the chances of getting funded for projects as well. So I'm just flagging that.
So this is my being a novice researcher, I guess, in doing data sharing like this, but is this something that's best done before we start collecting data, or have you had any luck in the past saying, hey, by the way, we would like to start contributing to this larger body and asking for a data sharing agreements like that kind of as it goes? I guess which
ideally it happens first,
right? It seems like I should go ahead and
after.
start doing this now before we start collecting data.
There may just be a line you need to add into your IRB basically that says something along the lines of, we intend to de-identify our data set as required by NIH, and we may share that, and it may be used for other analyses. It's really broad, like a blanket statement, but they may require that to go into your consent forms as well, so that subjects are consenting for the use of their data de-identified for other projects. I haven't seen this be a barrier. Carolyn may know more than I, but it's basically, it just needs to be in there, but it's not something that should raise alarms or cause major problems. This is definitely the direction that every major funder is heading. And it's just one line to the IRB.
I got it. Yeah, it sounds like it's something
and
I should.
It's ethical, because if you guard your data and it can't do the most scientific good, then you are sort of robbing the patient who agrees to participate of their potential benefit and impact. So it's totally standard. Your IRB should not have a problem as long as it's not DNA. That's a more sensitive topic.
Okay.
Let us know if you need any, I don't know if you get any pushback from the IRBs, but we all hate amendments, but basically we just submitted an amendment to our IRB to make sure that line was in there, and that was kind of it.
So I haven't yet submitted my amendment for the new Konica device either. So I'm about to submit a few things, and plus some of Carolyn's edits. And so that's all going to be dumped into one big, huge, hey, we got some changes to make. And so I'll just add that. I'll just make sure it's there.
Perfect. Thanks, Brady.
Yep.
I don't have anything else related to the data repository, but again, please add your comments or reach out to me and Carolyn if you have any thoughts on this, and we'll be in touch with updates in the next couple of months as we get a draft of this going. And we'll, if anybody's submitting IRBs or amending, please consider adding some of this flexibility language into your document. Thanks all. Thanks, Carolyn.
Thank you. I think those were the major issues that we had hoped to tackle. Is there anything, any issues that anyone wants to bring up with their study that we could help think through or share as challenges that we might work on as a group or add to an agenda at a later meeting?
No, I just want to say publicly here, I appreciate your patience. I know I've asked you a lot of questions just because I wanted to be in alignment with you guys, and I know the complicated nature of this research. So I realize I've fired off quite a few emails to you guys, so I appreciate your patience along the way.
We're really happy to field questions by email or outside of this forum if anyone has you know, any issues they're struggling with. We'd love to make it easier for people trying to get these off the ground.
Yeah, thanks, Brady. It's also helped us think through some of our protocols and the way we're doing things too. So helpful to us as well.
Okay, I think with that, we're gonna be able to wrap up. Thank you all for making time and for joining. I'm gonna stay on for a little bit with Ash and catch up on maybe some FDA updates. But other than that, I really appreciate everyone's time, and please do email us if there are things that we can help with or things you want to talk about at the next meeting.